are very, very high level, as was the request. So this is based on, really a very basic introduction into phase 2-3 clinical trials, like the phase 1. Bear in mind that there are so many different permutations of ways that you can design studies. And this will be really a look at what one might term a traditional view of a phase 1 study, or a conservative view, of phase 2 and phase 3 studies. But this is not gospel. This is not the only way to do these. In fact, nowadays, many, many studies are actually designed in very different ways to what's presented here. But it's broken into phase 1, which we've done, phase 2, which we're about to do, and then phase 3. But again, bear in mind that sometimes you can use phase 1 studies, adaptive designs. They can become effectively your phase 2. You can go straight from phase 1 to phase 3. Your phase 2 can sometimes be your pivotal phase, effectively be your pivotal phase 3 study, but it's a phase 2 study. So there's a whole lot of other scenarios or ways of looking at this, but this is just really the traditional. We've done phase 1, we're looking at phase 2, we're going to look at phase 3 as discrete entities. Okay. So, oops, sorry, wrong button. Okay, what I've done is I've done the presentation in the same format as the phase 1, so they sort of all sit together. So the first things that we're going to look at is, or the thing that we're going to look at in the presentation is, why do we do the studies, what are the studies, and when do we do them? And then also the results. And the results in terms of, from a phase 2 perspective is, do we go? In other words, do we progress the compound into further development in patients? Okay. Do we not go? So is there additional information that we have to do? And that's the try again bit. The try again bit is, do we decide that this product isn't going to go any further, or are we going to go back, invest, do some more, perhaps it may be more phase 1 work, it might be more chemistry on the product, it may be going back into toxicology. But are we? So it's either we're going to go ahead into clinical development, we're going to kill the compound off completely, or we're going to refine the compound and then decide how we move it forward. So those are really the sort of key outcomes. So again, what are we talking about in phase 2?
Is Raymond blowing up?
Sorry. No. If I can. Okay. Thanks, Nick. Okay. So what are the phase 2 trials? And phase 2 trials are typically the first in-patient trials. So it's the first time that you're taking your product into the patient population. Again, just going back to what we said before, if you have an adaptive phase 1 trial, you may well have already got data in patients, but typically and conservatively, this will be the first time that you're doing a trial in your target patient population. There are some other riders. So in oncology, often your phase 1 studies are in patients straight away. If you have what's known as a high-risk molecule, your phase 1 may be in patients straight away. The point here, though, is that usually these are your first in-patient trials in a particular target population, or sometimes target populations. You've got more patients here than you have in phase 1, so typically between 100 and 500 patients that you're recruiting in. Again, that's very indication dependent. You may have many less than that. In fact, in some indications, if you could get 500 patients, that would be a phenomenal phase 3 program. But again, we're talking typically, let's think about an antihypertensive drug or something like that. That's what we'd be looking at. And really the outcome in phase 1, as all of you I'm sure remember, the outcome in phase 1 is safety. Thank you.
God, yesterday I told you efficacy.
Now we're looking at, we're interested in knowing whether there is actually a response of the drug in patients. So we are going to begin to look at efficacy endpoints. The important thing here to note, though, is that even in phase 2, we may not be statistically powered to truly show efficacy as such. What we're statistically powered to do is to show that there is an effective treatment. That's what we want to see. We want to be able to show that the treatment is having an effect. Okay, and that's quite important, because the way you usually set up the statistical analysis for a phase 2 study is you're saying, as I've said over there, we have these hypotheses. In statistics, the hypothesis that you're trying to prove is the null hypothesis. And here the null hypothesis is usually set up as saying there is no effective treatment in the patient population. And you're hoping that your statistics disprove your null hypothesis, that there is an effect, and therefore you can decide on how to take the drug forward into development. So again, the critical thing here is that you have a good statistician. Phase 1 studies, they're fairly standard in terms of the number of patients, the number of cohorts, the dose escalations that you're going to do. So they're quite standard. And although you do have statistics, you don't rely on the statistics in those studies. For a phase 2, you start relying on your statistics, so you do need to have good statisticians involved. These studies are often multi-centered, but they can also be single-centered studies. And what you're looking at here as well is the acute as well as cumulative toxicity in your patient. So you start looking at, you're still looking at safety, and you're still looking at acute safety as you are with your healthy volunteers. So, what happens immediately after dosing or within the first 24 hours of dosing, 4 hours of dosing, depending on the route of administration. But you're also starting to look at slightly longer term safety data. So you will have cohorts of subjects that you may follow for a little bit longer period of time to see, say, for example, what the 7-day, 14-day, or 28-day safety is in your patients. You still very much want to look at pharmacokinetics, because here you're looking at pharmacokinetics in the patient population, and that could be very different to the pharmacokinetics in a healthy population. So very, very important to understand that, and obviously understanding the pharmacokinetic, pharmacodynamic effect is absolutely critical. You'll be wanting to try and define the optimal dosing, optimal dose and dosing schedule. So at this point you may not know that in patients, so you may be trying out, you will be trying out different dose levels. You may be trying out different routes of administration. You may be trying out different formulations as well in phase 2. Again, possibly just worth saying that we're talking as though there's an individual study at each of these points. That's not true. You may have multiple studies. You may have multiple phase 1 studies. You may have multiple phase 2 studies, because you're learning and gathering information as you're going on. And you are somewhat limited by the flexibility in the protocol. So if you need to assess something additional and you haven't got that flexibility in the protocol, you have to close the study and open another one. So don't think of it as just being one phase 1 study, one phase 2 study leads into one phase 3 study. That's not necessarily the case. The other thing you'll also start assessing in much more detail now, if you have them, are your biomarkers. And the relevance of the biomarker to your patient population. And again, that's very important to tie in with your PK/PD, and your PD is often associated with that particular biomarker. Okay. The other point there is the KOL input is really critical in phase 2, and this is when you really do start to engage in terms of very close collaboration with KOLs. Up till now, there'll usually be a fairly loose association. KOLs may be giving input, they may be part of a scientific advisory board of the company or something like that, but now they start to get much more involved in the actual design, interpretation, etc. of data. So, sorry about all the bullet points, but just going into a little bit more detail about the what of phase 2 studies. The first thing to note is that they are more complex than phase 1. And they're more complex usually because you're trying to assess a number of different things. You're assessing patient safety or safety in patients. You're starting to assess efficacy. You're assessing biomarkers. You're assessing dose, route of administration, acute and chronic safety. So there are a number of different things that you're assessing, so they are more complex. You do require statistical calculation now because, as I said before, the big decisions that are being made here are, do we invest in a very expensive phase 3 program? Do we kill the compound, or do we need to go back and redesign or gather additional information? So this is really a pivotal point in terms of the development of a compound. So you need statistics because you want to be sure about the data that you're generating. But again, as I say, what you don't typically have here is you don't have a very robust efficacy assessment at this point in time. And the other point about this is that as you bring on, again, you're working within the confines of a clinical trial protocol. If you have to bring in more subjects into one of the arms, one of the doses versus another one, you may not be able to recruit into that dose level, so your statistical power in that calculation decreases. So you have to be very careful about how you look at the statistics around the phase 2 study. As I've mentioned already, there is no standard study design for phase 2, and I've put that in inverted commas because there's no standard study design really for phase 1 or phase 3. But some of the design in phase 1 is more standard, and by the time you're starting to do pivotal phase 3 studies, there are standardized elements of the phase 3 program that you will have to do. So there are standards of care that you have to show that your drug can influence. But in phase 2 is probably the area in terms of development, if you're having a look at discrete phase 1, phase 2, phase 3, where you can play around the most. Again, sometimes it's not within an individual, within one study. It may be that you need to do multiple studies, but it's a lot less standard here. There can be blinded or unblinded studies. The initial doses are based on information from phase 1. But bear in mind that if you've been able to do an adaptive phase 1, you'll be much more informed for your phase 2, your initial phase 2 dose levels. If you haven't done an adaptive phase 1 and you've done a phase 1 in healthies, then you may have more range finding work to do in phase 2 because your healthy population, the kinetics may be very different, the effect of the drug may be very different because the target may be up or down regulated in your patient population versus healthy. So you may have to do some additional work, but you'll have to base those initial doses on data that you've generated from your toxicology work, but also from your phase 1. So again, kinetics may be different compared to healthy volunteers. Adaptive designs are possible in the phase 2 space as well, and usually the adaptive design here may be that you may look at multiple different patient populations. You certainly will look at different dose levels, and what you try and do with the study design is you try not to be specific. You try and leave it a little bit open so that you don't have to go back and do revisions to your protocol or substantial amendments, which again the regulators don't like, and that influences your statistical power. So you try and leave some of the definitions in terms of exactly what you're going to be doing a little bit vague in the protocol so that you can stay within the protocol and still make these changes. And then, identification of the PK-PD relationship is really important in order to, be able to ensure that you're looking at the correct clinical endpoints from a PD perspective at the right dose level in phase 3. And writing it, I wrote that bullet point and I sat there and I looked at it and I thought, well, that's really nice and, it's one sentence, it doesn't take up much space. But that is so difficult to do. Even in the phase 2 setting, we still, you still have situations, and we've had them here at Crescent where we've had clients coming to us and asking us, whether or not we can do some assessment looking at a phase 2 program and seeing whether we think that the phase 3 doses are correct. And we have situations where there are competitor products to some of the products we're looking at where they've used multiple doses in phase 3 and the headaches that then bring later on in terms of trying to get the product into the market. So it looks like a really simple point, but it's actually very critical, but can be quite complex to actually do, and often very difficult in the biologic space where you may be dealing with needing to do loading doses and then maintenance doses, and so it is quite a technically challenging thing.
Can I ask a question, Raymond? When you're looking at dosing, are you only thinking about it as far as the clinical utility, or are you thinking about the cost of goods there? Are you thinking about feature payer decisions? Because we've had a number of instances in workshops where the dosing has a big impact on its commercial attractiveness.
Usually at this point, Jamie, the reality is it's usually driven by the clinicians and the scientists and not by the commercial teams. So in reality, I think generally speaking, it would be purely based on dose-response relationship in terms of getting the optimum dose. But yeah, and then usually it's the commercial team that has to try and pick up pieces and make a commercial go of what is perhaps a little bit of a mess. So yeah, I think mainly medical and scientific.
Yep.
Moving on into the why. Why do we do these? Well, we need to assess obviously the effects of the drug in patients. I mean that's obvious, it has to be done. We have to understand the PK/PD relationship in patients because we're going to use those data to define the phase 3 program, what parameters we need to look at, when we need to look at them, based on the dose of the drug that we're going to be administering. And so, you know, that's absolutely critical for the phase 3 program. But also, it gives us an idea now that the other reason why that's really important is if we can see in healthies that PK/PD was great, but in patients, the PK/PD relationship is not as expected or is giving us an issue where we now have to dose. In patients, we find we need to give them an oral dose 5 times a day. Nowadays, nobody's wanting to take an oral tablet 5 times a day. Then that would be one of those points where we may say we need to rethink this whole program and take it back to the design phase, see if we can create an extended release version or whatever the case may be. Patient safety and tolerability. This is generally our first assessment of safety and tolerability. Again, we're not going to push forward into a large phase 3 program if we have a significant safety signal, if we have something that worsens a comorbidity in our disease population. And also, at this point, we may have a situation where in healthy volunteers who have, say, a normal immune system, we don't have any injection site reactions, but in our disease population who may have a hyperactive immune system, if we're trying to give them an antibody to treat allergies, and they have significant reactions to that, local reactions to that, we know the drug isn't tolerated in our patient population, we may need to go back and reformulate or rethink about how we're going to be dosing this drug in patients. And then obviously, you know, we're looking at what the dose level is, the frequency and the route of administration. Again, that will give us an idea as to whether we need to go back and redesign, or just gives us an idea of, not an idea, it gives us the information required to set the schedule, the regimen, the route of administration for phase 3. So yeah, it's all about those 6 rights: the right patient, the right drug, the right dose, the right route, the right time, and all done with the right documentation, which is really important. And it looks so simple when you put it up there, but isn't necessarily. Sorry about that. Don't worry about the pictures. You've seen the slide already from the phase 1. I'm sure you all remember it very clearly. Just to give an idea, phase 2, as we were discussing yesterday, yeah, typically they're around 6 months or so in duration, but again, very much depending on your indication and depending on the number of different parameters you're trying to look at in your phase 2 program, can go up to 18 months. Another point just to mention, as I've said earlier, is that the phase 2 study in certain indications can actually be your pivotal registration study. So the results, what do we need to know? So the go button is pressed if the alternative hypothesis is valid. In other words, we are seeing a response in our patient population. We are, you know, able to clearly define that patient population. The dose level, frequency, the route of administration are all identified or at least significantly narrowed. So there can be a situation where you could go into a phase 3 with 2 different routes of administration. Very unusual, but it can be done. And then you have no significant safety signals that you're picking up in your phase 2. And something I've added in there that probably requires a little bit more discussion than just simply TPP supported. But if you think about some of the discussions that we've been having over the last few days here in Europe and that you've had in India already, one of the points of reflection or refinement of the TPP, or actually the genuine definition of the TPP, is around the end of phase 2 when you're thinking about going into phase 3. So you'll have He'll have your aspirational TPP, if you like, up until that point. Now you really do have data that you can plug into there. And what you'll be wanting to do is you'll be wanting to do some sort of commercial assessment. And as Jamie, as you pointed out, some of the commercial assessment may be somewhat damaged by the data from phase 2. But you want to know that this still has commercial potential in order to be going into phase 3. In terms of a no-go, obviously there the null hypothesis is valid. In other words, there is no response seen in patients. The drug is not tolerated in your patient population at the dose that allows for a reasonable response. So you may be able to get a response, but if all your patients are vomiting or suicidal ideation and that sort of thing that you don't think will be such that the product is in fact commercially viable, then you may elect to stop. Where you've not been able to clearly define the dose route, or the defined dose route is not tolerated, that would be another reason for a no-go decision, and that the TPP is just not supported by the data. So in other words, you'd either have to go back and reassess the commercial viability or redesign the product, because you may have, with the TPP, you'd usually have a minimum sort of accepted target product profile in order to be able to proceed. And if any of these data are suggesting, even if they may show some benefit, are suggesting that you wouldn't meet that minimum, then you'd probably want to go back and reassess. So just in summary, it's the phase 2 study is your study in a relevant patient population. You do it to assess the biological effect in the patient population, safety and tolerability in the patient population, to get an assessment of the PK/PD response and the dose level, frequency and route in your target patient population. And it's done after phase 1, or even, as I said, even possibly as part of the phase 1 if you've got an adaptive design. Okay. So that was a very rapid, high-level run-through of phase 2 studies. Any questions in terms of phase 2? And again, please don't consider that to be a comprehensive overview in any way, shape, or form. It isn't. But any specific questions anyone might have?
Is there any sort of major differences between small molecules or between biologics or anything like that, or is it always the same sort of process?
In general, the process is very similar in terms of what you're looking at. It's the timings that may be slightly different. So, say sometimes with small molecules, you would also be very, very interested at this point in looking at, when I say PK/PD, really what we're looking at is ADME as a whole. And so you'd be very interested in your metabolism of the product in phase 2. So there are certain elements that you would be really wanting to consider. You also look at that in phase 1 in healthies. Less of an issue for biologics because you don't really have that to look at. The other side of it, though, as I said, is sometimes in certain indications you would perhaps potentially even skip the classic phase 2. You go from phase 1 to phase 3 because your phase 1 is looking at your healthy at your patient population. And sometimes with biologics, you would do that because there are instances with some of the biologics, they're highly potent, and you wouldn't, for example, want to give an anti-VEGF to a healthy person necessarily. There was a time where you may not have wanted to give the biologics to healthy people who may be at risk of developing a cancer that would be used to treat at a later stage, so people who had significant family history or something like that. More and more, though, we are beginning to see biologics being used in the phase 1 space in healthies because the risk of immunogenicity, etc., is relatively low. But those are sort of, I think, the main areas where you may see some differences between small and large, but large molecules. But generally speaking, the sort of things that you're looking at are going to be fairly similar.
And failure rates between the end of phase 1 and the end of phase 2, and is there a rule of thumb there?
Again, really, really difficult to sort of say that there's an absolute rule of thumb in terms of failure. Failure is I would say less high in phase 1 and higher in phase 2, and the reason obviously is that you'd only fail in phase 1 really if you can't get the drug in, you can't get any exposure for whatever reason, or that you have a significant safety signal, because you're not looking at efficacy. But as soon as you start to bring efficacy into that equation, you have a higher risk of seeing a potentially negative result. So phase 2 attrition is usually higher than phase 1 attrition.
I mean, but we're talking about what?
60%.
60% failure in phase 2.
In small molecules.
In small molecules,
Small
yeah, absolutely. I mean, large molecules, that's one of the other, nice things about large molecules is that, as long as you are interacting with your target, again, because of the way you're administering it, and because you don't have metabolism issues to worry about additional safety concerns, if you're hitting the target, you're likely to have a physiological effect.
So what's the rate in large molecules?
That'd be much lower than that. I think you're probably looking at around 20% maybe, and then it's often due to formulation issues or stability issues or immunogenicity, and then often you can go away and have a look at modifications or purification or process to bring it forward. So that's, I mean, often you do struggle a little bit, because sometimes you do have to have an initial loading phase and then a maintenance phase to try to understand what that loading phase should be, how many doses you should give, how far apart they should be. That can be quite complicated with biologics.
Usually you fail the molecule rather than the molecule fails you because you got the wrong dose.
Yeah.
I think the other difference, major, I used to find in some of those benign antibodies, is that you never reach a maximum tolerable dose. You actually reach maximum costliest dose.
Yeah.
So you stop because you can't afford to do the trial.
Yeah. Okay, so we'll rapidly move into phase 3. Same disclaimers as for phase 2, for the phase 2 presentation. And apologies, some of these slides you will have seen in the previous one, but they were 2 separate decks that are now one. So sorry, I didn't have time to rationalize it as much. So we'll look at the what and the why and the when again, and then the result here is hopefully to make lots and lots of money. Because really what you're hoping to do here, of course, is to actually get a registration and get the product into the market.
Not to treat patients, but make a lot of money.
Of course, to improve the quality of lives of patients.
That is only on the annual statement.
But obviously the other result is potentially to try again. And I put in very small writing, which nobody will be able to see, and the development team gets it. So if you get to a phase 3 and you fail a phase 3, I mean, that is a big deal for the internal development teams. That's not a nice position to be in. Yeah.
That's when you have suicidal ideation.
That's when you So here, what we're talking about in phase 3 is we're talking about the registrational studies, or study in the first indication, or the first indications, depending on the plan of attack in terms of the strategy. But for simplifications, let's just say it's the registrational study for the first indication. So this is the study that you're going to do to generate the data, to take to the regulators to say, you need to give us approval for us to market this product commercially. Okay. So it is the pivotal registrational trial. They are most frequently multi-centered, and the reason for that, of course, is that they need to have large patient numbers. So we're talking, I've said they're greater than 500 because the previous slide, the phase 2 was 100 to 500. These can be in the thousands of patients. You could say, well, why do you need that to show that? You don't need that from a statistical perspective to show efficacy, and that's true, you don't. But what you also have to show is you have to try and show that you have a representative cross-section of the genuine patient population from a regulatory perspective. And of course, the more numbers you have, the greater your statistical power, and the regulators do want to see that. So the results from the primary endpoints are the basis for approval. Safety concerns here could lead to black box warnings, and that, of course, from a commercial perspective can be challenging. It's not the end of the world. A lot of very, very successful drugs have black box warnings. Humira is a good example. So it doesn't mean that there is going to be commercial breaks on, but it can be a challenge if there is a black box warning. Certainly can be a challenge if your competitors don't have one. The studies will result in your labeled indication the dose level on the label and the route of administration on the label. So, actually on the face of it, the phase 3 studies in some respects appear simpler than phase 2, and the reason is that you should by now have a very defined plan, whereas in phase 2 you may still be playing around with things a little bit. But you have to have very careful statistical calculation in order to ensure that your primary endpoints are correctly powered. If you don't have that, and you miss your primary endpoint, but it's because you haven't included enough subjects for how sensitive that primary endpoint is, unfortunately you have a failed study, and all your other endpoints just simply become observational data. Okay. So the study is designed to maximize the chance of success without introducing bias. I say that's sort of with a bit of a grin, because obviously when you're doing this you're spending a lot of money, so you are certainly going to want to maximize the chance of success, and it is likely that there will be an introduction of some bias. You will select the best possible patient group from within a representative patient group. You will hopefully have the optimal dosing regimen. If you're having to do a comparator study, you'll be trying your utmost to ensure that you're using the comparator within the specification of the comparator use, but at the worst possible way of doing that. So there are ways that you need to look at the study design to maximize the chance of success, because there is such a huge investment here. Obviously you still have to have the clinical and scientific data to support that, so you can't go and design a study that is absolutely going to skew everything in your favor, because the regulators will pick that up. Again, these studies are usually blinded and placebo-controlled, although they can also have an active comparator arm, which is becoming more and more important from a regulatory perspective, especially if you're not going into a disease area that's has a completely unmet need. So if there are products in the market, you would be expected to provide data showing your product versus the gold standard. And I say they're usually blinded and placebo-controlled, but there are instances in the oncology setting where it would be unethical to do a placebo-controlled study. So you would do a controlled study, but it wouldn't necessarily be placebo-controlled. And then the doses and route of administration will have been defined from phase 2, hopefully. The kinetics, you can still look at kinetics in the phase 3 study, and often companies do still look at kinetics in phase 3 studies, but very often they're what we call pop PK, or population PK, rather than individual PKs. So you'd be taking time points from potentially different individuals across the course of the study, rather than getting a profile from a single individual. And that, or you would be using a single individual, but from different regions, different areas, so that you can pull together. It's a much more diverse group that you'll be looking at the kinetics of, just to again give you more information on the kinetics of the product on a more sort of population level rather than the discrete level. And you wouldn't usually base decisions, go-no-go decisions, or reformulation issues, etc., based on pop PK data. And then, say, adapting the phase 3 design is usually not permissible. Okay, and so what that means is that if you miss a primary endpoint, you're basically going and then presenting observational data from then on. You can't go and say, well, a lot of clients, a lot of companies do post hoc analyses and show that in this patient population actually it may have worked, etc., etc. You can do that and present those data, that's fine. You can't use that in a submission. You can, that's really interesting scientifically, but you cannot go, once you've set your statistical analysis plan, if you make a change to that during the course of the study, the chances are the regulators will throw the entire study out. They really don't like it if you do that, because that could be then seen to be influencing the analysis of your data based on data that you're generating. So that is a huge no-no. So adaptive designs in phase 3 is not going to happen. Right, if everybody could just read the top
Sorry,
left.
Ben,
Sorry,
you can add secondary endpoints into your phase 3.
Yes.
I mean, just your registrational design, you
Yeah.
can't change.
yeah, absolutely.
So it's a
Sure.
difference,
Your
right?
secondary endpoints are
Phase
the
three.
endpoints that you're
Of adapting
you're
the phase
using to
design.
provide additional commercial data, etc., etc. Your primary, of course, is what you're going to be basing the label
Yeah.
on. So yes, you can do that, but you can't then change. Say, well, we're going to add a couple of secondary endpoints, and therefore we're going to change the way we're going to look at the statistical analysis of it. So the phase 3 studies are they're done because they're much closer to the real-world patient population and to clinical practice, and of course the endpoint here is a drug that's going to be in the market that physicians can prescribe and that patients will use. So you're getting much closer to that than the fairly tightly controlled phase 2 and phase 1 studies. That the claims on the label obviously have to be backed up by your phase 3 data, or should I say your pivotal data, because it may be phase 2. You're defining here the indicated patient population, the efficacy of the drug, and the safety and tolerability of it. All of that will have to go into the label. It's not really a why, but it's something to really think about for these sort of studies, and gives you a reason as to why they're so expensive. They're multi-center studies, and so therefore you're dealing with very complex logistics, which is especially important when you consider that although you're looking at much more of a real-world situation, you have to still have very, very tight control over particularly the drug itself, because it's not a registered product. And you know, if you're thinking about biologics, there's a whole cold chain issue that you have to deal with. So they're very, very complex. But then also, if you look at the data that's being generated, it's very complex in terms of you may have to centralize some of the assessment. If you're using central laboratories to look at blood parameters, you have to make sure that the central laboratories in the different regions are using the same sort of equipment, or that the machinery that they're using is calibrated in the same way. So they really are logistically very, very complex studies to run. And then critically, that statistical analysis plan. It's absolutely critical and it has to be locked down. You cannot change it. Unless there's a really, really good reason to, which the regulators typically say there is never a really, really good reason to. So how long do these studies take? They typically, 12 to 36 months. A lot of months there, many months. Again, it'll obviously depend very much on what your primary endpoint is, how long you have to dose patients in order to be able to achieve your primary endpoint measures. So that's why we look so carefully at the primary endpoint completion dates, because those are really critical. But bear in mind that the phase 3 studies can then also have long-term extensions onto them, very often for safety reasons. From our perspective, the primary endpoint completion dates, which is then gives the data on which the company can actually submit their registration, is critical for us and for our clients. So again, how do we base our decisions? The GO, which is effectively telling us that we can make our application for approval, is that we reach our primary endpoint. The safety is acceptable. So here again, we're not, we don't necessarily need it to be completely squeaky clean unless our TPP tells us we have to, but we have to have acceptable safety. The commercial model is valid. We get to the end of phase 3, we've got all our data, we're now going to make a submission to registration. We just have to make sure that the commercial model is still valid. In the 16 years that it's taken us to do our phase 3 study, has the market moved so that we don't want to bring the product to market anyway? We have everything required for a complete application, because bear in mind that the phase 3 data, although it's absolutely critical, has to be backed up with manufacturing data, etc. So we need to make sure that all of that is ready for us to be able to go ahead and make our application. We start now thinking also about readying commercial plans. Hopefully the commercial plans are ready, and that the teams are prepped for for launch, although, again, because of the approval process, a lot of the sort of recruitment of your commercial teams, etc., will only start to take place in earnest once the applications are in. You may start thinking about looking at setting up support services for the product if that would be required. A lot of the biologics need support services around them. You have to start thinking about pharmacovigilance plans and then obviously planning for additional indications. In terms of no-go, the primary endpoint is missed. Post hoc analysis may show a better target population. You'd still have a no-go decision at that point in time, but that could mean just a refinement of what you were doing, going back, doing an additional study, but in the correct target population. You just may not have defined it correctly the first time. So, will you do a follow-up phase 3 or do you need to kill off the compound? So, in summary, these are the pivotal registrational studies for the initial label. You do them to get a more robust assessment of the drug's safety and efficacy in the clinical patient population, and the data are required by regulators in order to make the correct risk-benefit assessment. So you have to give them data for them to be able to assess whether the product should be registered, and they have to do a risk-benefit assessment on that. So that's what these data are used for. And then it's typically done after phase 2, but again, or possibly even after an adaptive phase 1, if one's able to do that in an accelerated fashion. And so that's that. So apologies for going through that so quickly and very simplistically, but are there any questions on phase 3?
Yeah, about the SMB, the data safety monitoring. Can they stop? I mean, if the safety, if the data that they gather during the trial show very good result, can they basically stop it in the middle if it actually warrant that basically you can file with it?
No, you need to continue safety monitoring all the way through.
All the way through?
Yeah.
Right, because we had an investor call in one of the companies was saying that the MSC, the DSMB basically could stop the trial in the middle if they basically think that the data were enough to warrant.
No, they'll stop the trial for futility, but they're not going to stop the safety monitoring.
Oh, right.
Yeah,
okay.
yeah. Oh, yeah, absolutely. Futility, but you can't.
Okay.
Yeah, safety monitoring.
Yeah.
We will continue.
Yeah,
Okay.
you can.
Are there any idiosyncratic phase 3 requirements for individual markets? Like does the US require something that Europe doesn't, or vice versa, or whatever? Or is it always a pretty standard approach?
I mean, there certainly are differences in terms of how critically they may assess the data, whether it's a new technology or not, as Rakesh was saying the other day. But I'm not aware of any genuine idiosyncrasies that are such that they'd be so different. Because don't forget, a lot of these studies, the phase 3 studies, are done, at multi-centers throughout the world, all sorts of different regions. So you've got multiple regulatory agencies that have already viewed the data of the product up to that point in time, and there may be some slight tweaks from an ethics perspective that a company may have to then include in a trial for a particular region. But I think globally at that point in time it would be very unusual for there to be a specific regulatory requirement that would fundamentally alter.
But the Japanese do insist on the phase 3 in a Japanese population.
Sure. And China, you can't take clinical material out of China. So assessments all have to be done within China, human clinical material out. So yes, there are certainly those. There may be certain ethnicity requirements for certain regions. But yeah, I mean, Japan is the perfect example where you do have to have Japanese patients included in the phase 3 trial in order to get registration in Japan, or patients of Japanese descent, which is why there are clinical trial units in Hawaii.
In Hawaii, yeah.
Which I never got to go and inspect. It was really disappointing.
In terms of patient numbers, can the company increase or decrease the patient numbers when the trial has started in phase 3?
Ooh. If they can show that there was a mathematical error, and that their statistical analysis is not impacted. They're doing exactly the same thing, but they made literally a calculation error and so needed to increase, then yes. Or if it's something like a biosimilar and there's been a subsequent request to increase the patient population because a certain safety parameter, the regulators don't believe the power of the assessment there is sufficient, they may request a higher number of patients be recruited. So the regulators can request that. If a sponsor is doing it off their own bat, to increase the subject number probably wouldn't be an issue from a regulatory perspective. The regulators will always be happy with more data, that gives them much more confidence. To try and reduce the number, I think, would be a significant issue, but I think either way, you would need to have a very, very close discussion with the regulatory authorities regarding a change in your patient number, and I think it would typically be because there's a safety issue that you may have, well, there could be a calculation issue, or there may be a safety issue that you haven't adequately powered for.
Do you have an example in mind that you'd like to share?
I've seen some trials where there's an increase in patient number, but I'm not
That's
sure.
what I'm saying.
Yeah, but whether it's phase 2 or phase 3.
Okay, phase 2 you often can. You may need to increase patient number because you're bringing on another dose level or the route of administration.
I know what you're saying. Probably sometimes you see that in European clinical trial registry you see 342 patients, and when you go to clinicaltrial.gov you see 450. So that's just probably the way it's put up.
Yeah.
But once phase 3 is done, signed, the advice is not to touch it.
No, I think
Yeah,
Amar is
see, that's
referring to
IPF trials,
IPF,
yeah.
where they had to
So
change the phase 3
but
to
that-
include additional arms.
No, so that the regulators must have requested.
Oh yeah, yeah.
yeah.
The
So,
regulators.
Sure.
so you don't initiate that conversation. Basically, that's the death nail. It's all
Yeah,
right.
then you'd get to the point and they turn around and say you haven't got this patient
So
population.
regulators, if a regulator requests it, yeah, you have to do it.
Sure.
But when I regarding that trial actually at a conference with some KOLs, and what happens is that they refuse to include the patients in the trial because the comparator arm was placebo, rather than, because the thing is that there is not necessarily like center of care with like the drugs that they wanted to include as comparator arms, but like as there is not real cure for it and everything, so obviously KOL still wanted to include those drugs in the arms. So what happens is that they just refuse to get any patients in. And then they stopped the trials for about 6 months to get it included in new arms.
Yeah.
So it came more for like from the physicians who said no.
Interesting.
And can I ask a blonde, fluffy question, which is, for you personally, what's the most exciting phase? I know you're going into phase 1 now after this, but from your personal history, what's...
I mean, I think for me personally, the most exciting phase is actually an adaptive phase 1. That would be the most exciting thing to do, and the reason for that is that I have that scientific interest and also the clinical interest, and that's the first time you're really starting to generate human data on what your drug is. If I was a commercial person, I would be absolutely certain I'd be sitting here telling you that phase 3 is the most exciting, but for me it's phase 1. Adaptive, if I can start to get an idea of what the drug is looking like in patients, that to me is a really exciting phase. Being able to say, right, we've had this great idea all the time, but with some potential, but we don't really know that we've got a drug, and you can't really say you've got a drug until you've been into human. So
Phase 1 take about 15 years, if you remember, to generate. Could you have stopped that? Was it a design flaw?
yes and no, which is a terrible.
To generate, it was an antibody, wasn't it?
It's a wrong calculation. It was the affinity between a monkey receptor and a human antibody, which obviously was a couple of hundred or 2 logs lower.
Yeah.
So they used that dose, and obviously human receptor binds more tightly to the human antibody. So it's
monkey,
exactly, monkey.
yeah.
So therefore they got the dose wrong by 100 times.
What happened? What really happened was there was a whole element of complacency, but there was also a set way of calculating the starting human dose for drugs, but it was based on small molecules, and that's known as the no adverse effect levels, or NOAL.
Yeah.
And that was used in this situation. for biologics, you actually have to consider not just the level of drug exposure, you also have to consider how much interaction there is of the drug with its target. And in fact, in small molecules now, it's caused a change to where we look at the MABEL in minimum anticipated biological effect level in small molecules as well now. So in hindsight, it could definitely, and you know, we've done the calculations and you can see that they were 100 to 500 times higher in dose than they should have been. And you can do that all now with the calculations that we use, but so it has changed the way one does the calculations.
It changed the future things, yeah. If people didn't know what happened, there's an absolutely massive response to the drug, and a lot of the patients lost their fingers and toes, didn't they? I mean, it was a huge phase 1 up in Northwick Park in the UK, and these patients were incredibly sick, and they all got gangrene, and their fingers just dropped off, and all sorts. And yeah, it was pretty horrific. And
Well, it's basic lack of understanding of biologics, that's what it was.
Yeah. And people that then assume that biologics were inherently safe, that there wouldn't be any significant side effects because they're not chemicals.
Yeah.
And unfortunately, there was complacency, and that's what happened. People didn't, you know, they had the data, but they weren't assessing it in the correct way because that wasn't the routine way of assessing it. Now, of course, it's critical. Now we don't even worry with biologics. We typically don't really worry very much about toxicology studies at all. In fact, a lot of people are arguing that we shouldn't be doing any animal testing of, you know, standard toxicology with biologics because there's so little relevance to that, much more relevance to the cell-based assays, receptor-based assays, disease models if you have them. So.
Yeah, good.
So can I ask one last question? So after a phase 3 has finished and the patients think that they got a much more robust recovery rates and stuff, so what happens? Can the patient access the phase 3 drugs?
It depends very much on the company. So when you are designing your phase 3, you do have to commit to there's a basic level of support you have to give the people who are volunteering to be on your study. Usually the commitment is to provide the drug for the duration of the study. That's usually the commitment from the perspective. If it is a life-threatening disease, or if it's one where there are no other treatment options, the ethics committees can turn around and say, if the patient responds, you need to commit to providing them the drug on an ongoing basis, even if it's not registered, then it would be on a named patient basis, for a prolonged period of time. And the ethics committees will say we want that to be for the life of the patient. The pharma company will say we'll provide it for an additional 3 months or 6 months or something like that. Usually there's often there's a trade-off. There is no requirement to do that. There's no absolute requirement. You're required to provide study drug as long as there isn't a safety issue for the patient for the duration of the protocol. But there's no absolute requirement to provide study drug beyond that. But you know, a lot of companies are fairly compassionate from a compassionate use perspective to patients who've been on their trials, especially if they are deriving benefit. I don't know if you agree.
Oh, yes, yeah. I think this is a good PR exercise,
yeah.
Especially for mid-sized companies. I mean, compassionate use programs, even outside clinical trials, good PR.
Right. From a commercial point of view, if you have any fitness devices such as healthcare or Fitbit, what clinical trials would you recommend this to be included? Do you think phase 1 will be too early, or maybe the company should start thinking about introducing these devices in phase 2 onwards?
Oh, Ricardo. So for something like Fitbit, okay, how is that going to help me understand the safety of my drug? It's not. So phase 1, where we're only looking at safety,
Right.
I wouldn't use it there. But when you're starting to, if you're starting to look at the effect of the drug and say you're expecting increased effort tolerance, or you're expecting increased energy levels, or increased feeling of well-being, or something to that effect, then certainly I think something like Fitbit, which could monitor activity, is great. I think it would probably have more use outside of a phase 1 unit as well, because there people are monitored very closely. You could easily stick them on a treadmill or something like that, whereas that would be much more use for patients who are ambulatory out on their daily lives. And you may ask them subjectively, how are you feeling? Oh, great. So how far can you walk now? I don't know, a mile or so. You can check on their Fitbit how many steps they've taken and whether that's any different to before. The difficulty with things like Fitbit is, in order to then make any sort of claim on the data, you would have to have a validated mechanism of doing that, and Fitbit certainly is not a validated platform. So you could use it for observational data, you could use it to provide some interesting information, but in terms of it being a clinical endpoint at this point in time, that's not possible. But that would be great for them to go and invest in something like that, to create some platform that is much more validatable. It would need to be more accurate, etc. Then yeah, absolutely, it could be used for something like it. Maybe cardiovascular, the respiratory space. Somebody who's got COPD with very poor ether tolerance, you can give them a drug and you can show that via an electronic signal of some sort there's a completely subjective display of an improvement, which you can map and they can see that. That's quite nice. I would think from a patient perspective that would be quite nice to have. I would think it would be phase 2, phase 3.
One more question, Raymond. When it comes to data analysis, is it usually done by is it done by an independent body or is it done by the company representatives itself? I mean, obviously there will be a lot of bias.
As independent as possible, but what you usually find is that CROs are usually the ones doing the studies. They're usually, frequently it's their statistical groups that are doing the analysis as well, but they do try and keep them completely separate, and the important thing is the analysis is usually only done once you've got database lock, so you can't go back in and influence the data at all. You can do interim analyses. Again, those are not usually that frequent in phase 3 because you don't really want to. So what you'll do is you'll get your primary efficacy, your primary endpoint completion, you'll database lock the primary endpoint aspects, and then do an analysis on those and leave the secondary, if they're going for longer, and the long-term data points in the database open, but the primary endpoint database would be locked, and then the statistical analysis performed. And the statistical people within the CROs, for example, usually don't report into the clinical trials organization. They'll report elsewhere, or it'll be a bit of a tortuous reporting line into there, so that there isn't, you can't say, well, my manager was the person responsible for delivering a successful trial, is also responsible for my statistical analysis. That wouldn't be considered good clinical practice.
Thank you very much.
Thank you.
Much appreciated.
Fantastic, Raymond.